In the video Biological Parameters of the Laurel Wilt Pathogen, we learned of Raffaella aguacate, a seemingly close relative of the deadly Raffaella laricola, and its potential benefits in laurel wilt pathogen research. But how would scientists distinguish what makes one organism benign and the other so devastating? In order to determine clues to the mystery, the research turns to the molecular level and to the University of Florida campus, to the labs of plant pathologist Dr. Jeff Rollins, who is working to find key puzzle pieces between the two organisms.
One thing we have been able to do is work with this Raffaella aguacate, right? This is a non-pathogenic species of Raffaella that was first initially isolated from an avocado tree and subsequently has been isolated from ambrosia beetles.
Rollins says the difference is the basis of comparative biology studies, similarities in behavior making for valid comparisons that Raffaella aguacate can persist in beetles and avocado trees, but unlike Raffaella laricola, does not cause disease. Research takes place on two tracks. At the molecular level, genomes between the two species are compared. At the level where they can be more easily observed, Rollins' team has developed a labeling tool that improves visibility of the fungal pathogen so it stands out from other fungi.
So in the past, one of the difficulties of understanding the movement of Raffaella laricola inside the plant is we didn't have good tools for being able to visualize it, see it, in the absence of all the other fungi that may be in there, right? Some colonizing asymptomatically, others causing small amounts of disease. So one of the tools we've developed is the labeling of the fungus, of the pathogen, with a green fluorescent protein. And when we put this into the tree, right, we can follow it over time to see where the spores go immediately, where the spores go at 5 days, at 10 days, at 20 days. Etc. And so by doing that, we now have a better idea of what tissues within the tree are being colonized, and we know what form the fungus is while it's colonizing that tree.
And before the tree shows any symptoms. Rollins says the molecular track of his research suggests a genetic role that begins in a tree's xylem tissue.
We really need to be able to understand what factors this pathogen is using to colonize and to kill trees. And so if we can understand what the important factors or the processes, how the fungus is moving within the tree, which small molecules it's producing, what proteins is it producing, right? So if we can identify those factors, right, then perhaps we can exploit those factors, right? These will then, the strength of the pathogen will then become the weakness as far as control efforts are concerned. Part of this disease dynamic is the host response to it. At some point in time, the host recognizes something that this pathogen is producing, and it responds to that. And many times people think about it actually as an over-response. So it basically gums up its plumbing, right? It keeps the water from being able to freely move from the roots, up into the canopy.
Rollins hypothesizes this happens as some of the tree's genes power up while others shut down. He says there are no hard answers yet about how this knowledge will help growers, but that the ability to track the pathogen in the tree and know what form it's in, for example spores or hyphae, is providing information about pathways that would not have been possible in the confines of traditional Petri plate studies.
We're able to go in and sample and look at gene expression of the pathogen. So we know what specific genes are being turned on or turned off during disease development versus when it just grows in a Petri plate, in the lab. And so from that, one of the interesting findings that we recently published is that this pathogen is able to turn on alternative sulfur utilization pathways. So it appears to be under some type of sulfur starvation, or it competes with the host for available sulfur during colonization. So we don't entirely understand that and know what that means as to which sulfur sources are most important or how it might be competing with the plant for that. But we have been able to make mutants within that pathway, and we're really exploring how essential this, what appears to be perhaps a battle for sulfur with the host is, and ultimately in causing disease.
And that, says Rollins, may lead to ideas about ways to use sulfur nutrition to manage the disease. He says there are no hard answers yet about how this knowledge will help growers, but that they should feel assured that as the research progresses, results will help inform us on potential new control tactics for the pathogen.
If we could identify if there's a particular factor from the fungus that the plant is actually recognizing and causing that overreaction, then one thing we could do is we could isolate that factor. We could actually screen trees, other germplasm, for those that don't have an overreaction to that factor. So that's one idea about how, understanding at the molecular level can help us put something into action that would help us to identify something that we can bring directly to the field.
As laurel wilt continues to impact the Florida avocado industry, the research team remains dedicated in finding possible solutions. This includes control methods designed to help stem this devastating disease. In our next segment, we'll take an extensive look at the cultural strategies being explored to control and mitigate the effects of laurel wilt.